I welcome you to today's webinar and to the presentation about the hallmarks of cancer. My name is Stefan Pellenz and I will be presenting today's webinar. This webinar is being recorded and we will make it available on our website within the next week. Any personal information about participants will be removed, so don't hesitate to use the chat window to ask any questions that will be answered in the Q&A session subsequently to the presentation. After a quick introduction, we will discuss the hallmarks of cancer concept and the different cells. The different cell biology facets it comprises. Subsequently, I will discuss several of these facets in more detail and also provide a list of relevant antibodies available from antibodies-online pertaining to these. Before then concluding the webinar, we will shortly touch on a related concept, which is the hallmarks of aging, and there will be a Q&A session to finish things off. Organizer of today's webinar is Antibodies-online. Antibodies-online was founded in 2006 as a spin-off of the Aachen Technical University in Aachen in Germany. And we are now located in Aachen, Germany, as well as in Philadelphia and Pennsylvania in the US. We offer today more than 4 million proteomics products from approximately 250 suppliers, and we consider it our mission to make the search and purchase of antibodies, proteins, kits, and other proteomics products easy and straightforward. We count more than 6,000 institutions in all 65 countries as our customers, some of which are indicated here in the middle of the page. Through our independent validation program, we offer a full refund for life science products sold through our platform in exchange for validation report. So up to today, we have concluded more than 1,000 validations of our products in collaboration with more than 200 scientists. So this program is open to you. If you're interested in validating any of our products in your particular experimental setting, please feel free to get in touch with us at independent-validation@antibodies-online.com. A quick word about myself as today's speaker. My name is Stefan Pellenz, as I said initially, and I have obtained a Ph.D. degree from the Université Paris 7 in Paris, France, for work on mobile genetic elements and DNA damage repair pathway choice in yeast. During my postdoc at the University of Washington in Seattle, Washington, I worked on genome engineering in mammalian cells using various endonuclease platforms. I am now working at Antibodies-online as a product manager, and I take care also of our customer validation program, which I had just mentioned on the slide before. So with the introduction being concluded, let's proceed to the hallmarks of cancer. In spite of the comprehensive term cancer, cancer is not one single well-defined disease. Actually, different forms of cancer can have indeed very diverse causes, affected tissues, treatment options, as well as different prognosis. However, in spite of their genotypic and phenotypic differences, cancer cells share certain mechanisms that enable tumor development. These common characteristics, termed the hallmarks of cancers, are observed in many types of cancer and are thought to be promising targets for cancer therapy. Understanding the role of the different hallmarks of cancer, their underlying causes and common characteristics can inform the development and new preventive and therapeutic approaches. The hallmarks of cancers were first conceptualized in a publication in 2000 by Douglas Hanahan and Robert A. Weinberg at the UCSF and at MIT respectively. It was then updated and extended in two further publications in 2011 and most recently last year in 2022. In this latest iteration, the hallmarks of cancers contain the following biological capabilities of cancer cells frequently acquired during tumor development. Sustained proliferative signaling, evading of growth suppressors, non-mutational epigenetic reprogramming, avoiding immune destruction, the ability for limitless replicative potential, tumor-promoting inflammation, polymorphic microbiomes, deactivating invasion and metastasis, sustained angiogenesis, senescence of cells, genomic instability and increased mutation rates, the ability to resist cell death, deregulation of the cellular metabolism, and finally the unlocking of phenotypic plasticity. I will shortly touch on those hallmarks marked in red here on this slide. I will point out one significant signaling pathway for each of these, and so including the significance of the respective pathway in healthy cells, some of the key actors for each of the pathways, as well as how alterations in these pathways can lead to cancer. I will also suggest several noteworthy antigens for each of these signaling cascades and a selection of antibodies-online products related to each of these antigens. These product lists are not exhaustive, so please feel free to get in touch with us to inquire about additional products that I won't mention. Cancer is a vast research area, as you surely are aware, and this presentation does not exhaustively cover all hallmarks of cancer, but it will hopefully be helpful to develop an understanding of some of the underlying causes. So with that being said, let's get started and dive into the first hallmark of cancer. So the first hallmark is sustaining proliferative signaling, and in context with RTK signaling here. The propensity of cancer cells to grow uncontrollably is probably one of the first things that come to mind when thinking about cancer. One of the predominant cellular signaling pathways is the RTK signaling cascade, and it is heavily involved in determining a cell's growth. RTK, or receptor tyrosine kinases, are membrane-bound kinases that are activated upon binding of receptor-specific ligands. These make up the largest class of membrane receptors that trigger signaling cascades through their inherent enzymatic activity. Their structure, activation mechanism, and key components of the signaling pathways are highly conserved in metazoans. There are in total almost 60 known RTKs in humans, which are grouped in about 20 classes depending on topology. RTK signaling involves the activation of a transmembrane receptor by the binding of a specific ligand to its extracellular domain. The activation triggers then the autophosphorylation and intracellular tyrosine residues on the receptor, which then act as docking sites for downstream signaling molecules that initiate cascades of intracellular signaling events. Despite the exceptionally large variety of receptors and outcomes, RTK engage only a rather limited set of core processes. RTKs are essential for crucial signaling pathways like NF-kappa B, PI3K-ACT, MAPK, JAK-STAT signaling, as well as RAS signaling. Accordingly, they relate to many hallmarks of cancers, including but not restricted to the sustained proliferative signaling that I put them into here. So the main reason for me to put them into this omnipresent signaling pathway in this particular hallmark is the inclusion of well-known growth factor in receptors, namely the epidermal growth factor receptors, EGFR, including HER2/neu, the insulin-like growth factor 1 receptor, fibroblast growth factor receptors, and vascular endothelial growth factor receptors. RTK signaling plays a critical role in regulating cellular processes such as cell growth, and differentiation, migration, survival, and dysregulation of this and of the signaling pathway, and it has been implicated in the development of various cancers and other diseases. The table on the right here on the slide lists a few antibodies against the aforementioned RTK growth factor receptors that are frequently overexpressed and therefore valuable therapeutic targets. As I have mentioned a moment ago, there's a plethora of RTK, so please contact us if you're looking for antibodies against a particular receptor not listed here. Well, those listed here obviously as well. But, so the next hallmark of cancer then is the limited replicative potential. Differentiated cells typically have a limited number of cell divisions before they die. This inherent characteristic is referred to as the Hayflick limit. Embryonal cells, however, do not senesce or age in the same way as adult cells do. They are in a state of rapid growth and division, which has a striking similarity to cancer cells. The hedgehog signaling cascade shown here is a highly conserved developmental pathway that plays a critical role in regulating cell proliferation and differentiation, tissue polarity, and patterning during embryogenesis and tissue homeostasis. It is silenced in most adult tissues. Activation of hedgehog signaling in cancer cells, however, can promote the expression of genes that are involved in cell proliferation, survival, and self-renewal, leading to the acquisition of limitless replicative potential. Different hedgehog ligands, including homolog sonic hedgehog, SHA, Indian hedgehog, IHH, and desert hedgehog, DHH, are produced as precursors. Release and extracellular accumulation of these mature ligands is regulated by the homologs of the Drosophila dispatched or DISP protein. Binding of secreted hedgehog ligands to the receptor homologs patch1 and patch2 triggers the stimulation of the signaling network shown here in the image on the right half. The repressive effectors of patched on the transmembrane receptor smoothened or SMO is thus relieved, which leads then to the activation of glioma-associated oncogene or GLI transcription factors. A key specialized structure in this entire process is the microtubule-based primary cilium. In the absence of the hedgehog signal, unprocessed non-activating GLI proteins are as well as their regulator SUFU are concentrated in the distal tip of the primary cilium, as shown here on the left side of the in the figure. Upon binding of the hedgehog ligand to patch, smoothen translocates to the primary cilium and SUFU is degraded then. The GLI transcriptional activators can then activate transcription of genes contributing to a cancer cell's proliferative potential. Aberrant activation of hedgehog signaling has been implicated in various types of cancer, including basal cell carcinoma, medulloblastoma, and pancreatic cancer. Targeting hedgehog signaling has become a promising strategy for cancer therapy because of this. So with several drugs approved for clinical use that inhibit components of this pathway, such as smoothened or the GLI transcription factors. However, resistance to hedgehog inhibitors can arise through various mechanisms, such as mutations in a targeted receptor or its downstream effectors, or activation of alternative signaling pathways. Antibodies-online products against many proteins that play a role in hedgehog signaling, like, for example, the hedgehog ligands, patched or smoothened, the SUFU regulator, and GLI transcriptional activators, important for intracellular signaling, are shown here on the right with a couple of representative products. Again, we have more, so if you are interested in products that are not listed here, please get in touch with us. So the next one then is the unlocking of phenotypic potential. And just like proliferation and replication, the phenotypic potential of differentiated cells is limited naturally. In contrast, unlocking phenotypic plasticity in adult cells is a typical characteristic for cancer cells. The Wnt signaling pathway plays a major role in regulating cell differentiation during development. The name Wnt is derived from the combination of the segment polarity gene wingless, or WG, from Drosophila, and the proto-oncogene INT1, which has now been renamed to Wnt1. It alludes to the role that Wnt plays in regulating cell proliferation, differentiation, and survival. Its dysregulation has been linked to various types of cancer, and Wnt signaling has consequently been implicated in several hallmarks of cancer as well. In the canonical Wnt signaling pathway shown here on the left, binding of Wnt protein to Frizzle 7 transmembrane receptor triggers a signaling cascade. A key component of the canonical Wnt signaling pathway is beta-catenin, a co-activator of the TCF-left family of transcription factors. In the absence of Wnt signaling, shown here in this image on the right, beta-catenin is phosphorylated by a complex of proteins known as the destruction complex, consisting of the scaffold protein exin, the tumor suppressor protein APC, or adenomatosis adenomatous polyposis coli, the casein kinase 1, and glycogen synthase kinase 3 beta, or GSK3 beta. Phosphorylated beta-catenin is then targeted for ubiquitination and is degraded by the proteasome. Activation of the canonical Wnt signaling pathway, shown here on the left, leads to beta-catenin stabilization, thus then its nuclear import and the expression of genes involved in cell cycle progression and proliferation. Mutations in this pathway can lead to constitutive activation of beta-catenin, and because of the role Wnt signaling plays in regulating cell proliferation, differentiation, and survival, such mutations have been implicated in various cancers, including colorectal, hepatocellular, and ovarian cancers. So not surprisingly, for the product list, the list of recommended products for research on Wnt signaling pathway is led by selection of beta-catenin antibodies, followed by some antibodies against Wnt ligands, and those against TCF-left transcription factors and components of the destruction complex to finish off the list. The list of antibodies represents only selection, again, of antibodies we offer against components of the Wnt signaling pathway. If your antigen of interest are not listed here, please feel free to get in touch with us. Which brings us already to the next hallmark of cancer, which is the evasion of growth suppression. Cancer cells are characterized by the ability to avoid growth suppression. That means normally proteins usually regulate cell proliferation by inhibiting cell division cycle progression. So it enables cancer cells, on the other hand, are unable to proliferate uncontrollably, thus leading to tumor formation. Under normal conditions, the cell division cycle is very tightly regulated by a series of molecular events that ensure proper cell growth and division. It is split up into 4 major phases based on events unfolding in the cell. During the first phase, the gap phase or G1 phase, proteins that are essential for DNA replication are being produced. Then during the following synthesis or S phase, the cell's genome is being replicated. And during the second gap phase or the G2 phase, you see a rapid cell growth as well as the production of proteins in preparation for the subsequent mitosis or M phase, in which the cell then ultimately separates. Cells that are not actively dividing enter a quiescent state in the resting phase, which is called G0. The progression through the cell division cycle is tightly coordinated by cyclin-dependent kinases or CDKs, as well as the activators, the different cyclins. Specific cyclin-CDK complexes persist in an active form only for a very short time, and they are then degraded by or inactivated by the time that a particular phase of the cell cycle is completed. The activation of a specific cyclin CDK complex is characteristic for different cell cycle phases, as indicated here in the cycle, and they drive the production of molecules necessary for the respective phase. Additional levels of dynamic controls are also provided by CDK inhibitors, which block the CDK function even in the presence of the requisite cyclins. 2 major cell cycle checkpoints assure the integrity of the genome and proper cell cycle progression. Once the DNA replication has been initiated, however, it must be finished. So therefore, the major checkpoint in the cell division cycle is the restriction or R point between the G1 and the S phase prior to DNA duplication. Progression beyond this point then depends on mitogenic and inhibitory factors such as DNA damage, signals from various signaling pathways, like, for example, Wnt, indicated here in the upper right. You can see here in the upper right that the control of these checkpoints involves regulators of very well-known regulators such as p53, MYC, FOXO transcription factors, and RB1. These are all proteins that are well known for cancer-associated mutations. p53 can also induce apoptosis, which I will cover in the next slide. The second opportunity then for arrest is provided between the transition from the G2 to the M phase in case of DNA damage. Loss of cell cycle control is closely linked to the evasion of growth suppressors. Cancer cells have the ability to bypass this control mechanism by inactivating growth suppressor proteins, thus enabling uncontrolled growth. As illustrated in this image, many genes and proteins are involved in the regulation of cell cycle division and growth suppression. Mutations in any of these genes can disrupt the cell cycle and thus contribute to cancer development. The product list shows a few representative antibodies for cyclin-dependent kinases, driving cell cycle progression and the respective cyclins. In addition, here there are a few selected products listed for the regulators P53, FOXO, MYC, and RB1. For any additional antibodies, proteins, or other products in the context of cell cycle control, please get in touch with us. The next pathway on our journey through the selected hallmarks of cancer is apoptosis and its significance for cancer cells' ability to resist cell death. Apoptosis, in general, is a programmed cell death pathway that eliminates damaged or unwanted cells in a controlled manner. It's a critical physiological process in multicellular organisms in order to maintain tissue homeostasis and to prevent cancer development. Understanding its dysregulation in cancer has led to the development of new therapeutic approaches. The equilibrium between cell growth and division and the rate of which cells undergo cell death allows for a dynamic adjustment of the cell number depending on internal or external parameters. For example, during the development of the vertebrate nervous system, about half of the cells undergo apoptosis shortly after they've been formed. And in an adult organism, the equilibrium is essential to maintain the size and function of organs and tissue. Dysregulation of this equilibrium oftentimes leads to cancer. Unlike necrosis, which causes potentially damaging inflammatory responses after affected cells burst open, apoptosis unfolds in a very organized way. So the cell shrinks first, and it condenses while the internal structures are being disassembled. The DNA is fragmented. The dying cell is then rapidly phagocytized by neighboring cells or macrophages. At the core of the apoptotic process are a set of caspases, which are a family of cysteine proteases that cleave target proteins to trigger cell death. They are produced as procaspases, which are rendered active subsequently to cleavage by other caspases. This caspase cascade is triggered when initiator procaspases, like for example procaspases 8, 9, and 10, are aggregated with the help of adapter proteins, thus facilitating mutual activation due to low protease activity or conformational changes of the procaspases. These then activated caspases are then free to activate further effector caspases like caspases 3, 6, and 7 to further promote apoptosis. The effect is then additionally regulated by the BCL2 family of proteins and the inhibitors of apoptosis or IAPs, like for example BERK1 or BERK4. Apoptotic processes follow several pathways depending on the death stimulus. There's the extrinsic death receptor pathway, shown here in this image on the left, which is induced through ligands that bind to a family of death receptor proteins, like, for example, TNF, FAS, or the trail receptors, containing a cytoplasmic death domain. The intrinsic pathway, shown here, mostly on the right, is triggered by intracellular signals. These can, for example, be DNA damage, which then engages the intrinsic pathways via P53, that I had already mentioned on the previous slide. Another trigger can be mitochondrial stress, which leads to the release of cytochrome C from the mitochondria. The cytochrome C then binds to APF1 to form the apoptosome, which activates caspase 9, and then which then triggers the downstream caspase cascade. In the cancer context now, triggering the intrinsic pathway in response to DNA damage is particularly relevant. Dysregulation of apoptosis can contribute to cancer by by allowing damaged cells to survive and proliferate instead of being eliminated. So, for example, some cancer cells may upregulate anti-apoptotic BCL2 proteins or downregulate pro-apoptotic ones to avoid cell death. Targeting the apoptosis pathway is a promising strategy for cancer therapy, with several drugs already in use or in development that aim to induce apoptosis in cancer cells. There are also caspase-independent pathways on the very far right here, which are triggered, for example, by coenzyme B and coenzyme A, And these caspase-independent pathways are thought to have evolved in response to viruses that inhibit that have the ability to inhibit caspases. The product list for the apoptosis-related targets contains antibodies against effector caspases 3 and 7 and initiator caspases 8 and 9. I also included a list of antibodies against the apoptosome components APAF1 and the apoptotic regulators BCL2, BERG1, BERG4. I did not list product as actually I did in the end. I did not want to list SKUs of antibodies against death receptors just because there are so many of them. But obviously we do have antibodies against TNF and FAST receptors as well. So please get in touch if you so desire. Next hallmark of cancer on our list is also caused on the cellular level by activation of caspases in response to external stimulation. We are talking here about inflammation and the inflammasome, the structure which is central to the inflammation on the cellular level. Inflammation per se is a body's response to injury, to infection, or to tissue damage. Different inflammatory pathways are triggered by specific substances like bacterial, cellular components, viral or bacterial nucleic acids, or pathogen or damage-associated molecular patterns, so-called PAMPs or DAMPs. And ultimately, all the different pathways lead to the release of pro-inflammatory cytokines IL-1 beta and interleukin 18, predominantly by activated macrophages. Under normal circumstances, this release results in recruitment of immune cells to mount an immune response, proliferation of fibroblasts to rebuild damaged tissue, and to phagocytic cells that engulf cellular debris or apoptotic cells. However, inflammation plays also a due role in cancer. On one hand, acute inflammation is a critical component of the body's immune response and can help to eliminate damaged cells that could give rise to cancer. But chronic inflammation can contribute to the development and progression of cancer in several ways. Firstly, inflammatory cells can produce reactive oxygen and nitrogen species, which can damage DNA and promote genomic instability. Secondly, excreted cytokines and growth factors can help create a microenvironment that is conducive to the growth and spread of cancer cells. And thirdly, chronic inflammation can aid cancer cells to evade detection and elimination by suppressing the immune system. At the center of the inflammasome activation pathway is the inflammasome, as the name kind of gives it away already. So this protein complex is a component composed of sensor proteins such as NLRP3 and an adapter protein like PCAR and the effector caspase Cas1. The inflammasome is regulated by the activation of the sensor protein, which can be triggered by various stimuli such as pathogen or danger-associated molecular patterns. And once formed, the caspase promotes maturation of pro-inflammatory cytokines, interleukin-1 beta and interleukin 18, and gastamine through proteolytic cleavage. Processing of IL-1 beta and IL-18 and gastamine D drives then pyroptosis, which is a highly inflammatory form of programmed cell death. Subsequently to cleavage, the N-terminal part of gastamine D localizes to the plasma membrane and then forms pores through which the produced interleukins 1 and 18 are released to further drive inflammation. So in summary, chronic inflammation can contribute to the development and progression of cancer by promoting genomic instability, creating a pro-tumorigenic microenvironment, suppressing the immune system, and facilitating tissue remodeling and invasion. The product list references selected antibodies against proteins forming the NLRP3 inflammasome. Antibodies-online does offer considerably more related products than these. Also, I focused here pretty much only on the NLRP3 inflammasome because it's the best studied. This, however, does not imply that our product catalog only covers this particular inflammasome. So please inquire as needed. Tissue remodeling, which can be added by chronic inflammation, as we have just learned on the previous slide, is among the other aspects important for inducing or accessing vasculature. Another pathway that is particularly important in this context is the Notch signaling pathway. Notch signaling regulates cell fate specification and patterning in vertebrate central nervous system during development, so mitogenesis, cardiovascular, and endocrine development. Its influence on inducing new vasculature is especially significant for cancer due to the high metabolism of cancerous cells and thus the need for nutrients. Notch signaling is somewhat unique compared to the most other signaling pathways. So for one, both the Notch receptor and most of its ligands of the DSL family are transmembrane proteins, which means Notch signaling always involves two adjacent cells. Secondly, the Notch signaling cascade does not include an amplification mechanism. Many signaling pathways rely on kinases that phosphorylate downstream activators, which then go on to trigger additional signaling cascades, such as PI3K, ACT, or NF-kappa B signaling. Even more, here in case of Notch intracellular domain, NICD in this image here, is then ubiquitinated upon binding of the transcriptional factor CSL. That means signaling is actually dampened instead of amplified. So this very tight control of Notch activity and the transcription of Notch target genes is crucial because of the Notch signaling pathway's impact on the balance between stem cell renewal and differentiation. Thus, its dysregulation has been implicated in promoting tumor angiogenesis, which is essential for tumor growth and metastasis. Notch signaling can induce the expression of vascular endothelial growth factor, which is a key regulator of angiogenesis and thus promote angiogenesis. That means the formation of new blood vessels in a tumor. Targeting Notch signaling has therefore emerged as a promising strategy for anti-angiogenesis therapy in cancer, as inhibition of Notch signaling can block tumor angiogenesis and decrease tumor growth and metastasis. Recommended products in context with this pathway are antibodies directed against the different Notch proteins and DSL ligands DLL1, 3, and JAK1. Other relevant proteins are Adam metalloproteinases, which are involved in Notch and Notch ligand regulated intermembrane proteolysis. And then the mastermind line of transcriptional coactivators for Notch. Like for the other slides, please get in touch if you're in need of products against any other antigens not listed here. So the deregulation of cellular metabolism and the rapid growth of cancer cell is the main reason why the vasculature is so important to sustain a tumor. I reiterated this several times on the last slide regarding Notch signaling. So the best known deregulation of the cellular metabolism in cancer cells is the Warburg effect. This effect was first described by Otto Heinrich Warburg in the 1920s, who remarked that cancer cells have a preference for fermentation of glucose to lactate, even in the presence of sufficient amount of oxygens. So that means that the cell relies mainly on glycolysis, and pyruvate is converted to lactate by lactate dehydrogenase, or LDH, in the cytosol instead of being transported into the mitochondrion to feed the citric acid cycle, which is a normal way of producing energy from glucose. This preference is somewhat counterintuitive since the aerobic glycolysis only produces 2 molecules of ATP per glucose molecule, whereas oxidative phosphorylation, so the Krebs or citric acid cycle, yields between 32 and 34 molecules of ATP per molecule of glucose. So, however, the Warburg effect provides a different selective advantage to cancer cells by providing building blocks, so nucleotides, amino acids, lipids, which are necessary for the rapid proliferation of growing cancer cells. Also, lactate is being transported out of the cells, which further contributes to the acidification of the tumor microenvironment. Because of the diversion of pyruvate towards lactate, glutamate becomes the main carbon source for to replenish metabolic intermediates in the mitochondrial citric acid cycle to cover the cancer cell's energy needs. Gain of function mutations in the isocitrate dehydrogenase in the cytosol, or IDH1 down here on the lower right, and in the mitochondria, IDH2, lead to the reduction of the alpha ketoglutarate, shown here as alpha Kg, which is one of the acid cycle metabolites, to 2-hydroxyglutarate, or 2-HG. This is an oncometabolite that can inhibit the alpha Kg-dependent deoxygenases by decreasing the concentration of this obligate cofactor. The class of alpha Kg-dependent deoxygenases then comprises various chromatin-modifying demethylases and methyltransferases. So the inhibition then leads to CpG island hypermethylation, and it can affect cell fate. Alpha Kg-dependent deoxygenases also include prolyl hydroxylases, which influence the activity of the hypoxia-inducing factor, HIF1, which is a master regulator of transcription in the adaptive response to hypoxia. Regulation of factors like HIF1, but also MYC, and alterations in the PI3K ACT signaling then further exacerbates the Warburg effect in cancer cells. And inactivation of tumor suppressors like P53 are also an important mechanism facilitated by the Warburg effect. So under normal conditions, P53 negatively regulates glycolysis and promotes oxidative phosphorylation. However, these mechanisms fail under aerobic glycolysis conditions, thus supporting continuous growth and survival of cancer cells. The two enzymes that are crucial for the Warburg effect are the isocitrate dehydrogenase in the cytosol and in the mitochondrium, so IDH1 and IDH2, since their gain of function mutation lead to the diversion and reduction of the concentration of alpha Kg. So accordingly, anti-IDH1 and 2 antibodies are found here on the top of the list of recommended products for this pathway. And the third antigen listed here then is the HIF1 transcription regulator, because of its significance and its impact on the expression of various enzymes depending on hypoxic conditions. I did also include antibodies against transporter proteins for the import of glucose and glutamine and the transport of lactate across the membrane. This list appears rather short because most of the processes which are part of the Warburg cycle are actually fundamental to cell physiology, like, for example, the lactate dehydrogenase, and involve proteins. So they're not listed here. If you're looking for products related to glycolysis or just the Krebs cycle or other aspects, and obviously also the Warburg effect, please let us know. So, and I have just mentioned in context with the Warburg effect, one of the consequences of this effect is the inhibition of chromatin-modifying demethylases and methyltransferases belonging to the alpha Kg-dependent deoxygenase enzyme class. This inhibition then can lead to epigenetic chromatin changes and ultimately result in altered protein expression. So epigenetic modifications include DNA methylation, altered transcription of non-coding RNAs, chromatin remodeling, chemical modifications of histone side chains. Dysregulation of any of these mechanisms then can lead to altered transcription and translation of oncogenes and anti-oncogenes, and they all have been implicated in various cancers. So for this presentation, I'm focusing on epigenetic histone modifications. Histone proteins form the protein core of the nucleosomes, which are the basic organizational unit of chromatin. Addition of certain chemical groups to the amino acids outside of the histone protein core impact packaging of chromatin. Specific patterns of chemical modification on histone proteins that regulate gene expression by controlling the accessibility of DNA to the transcription machinery is referred to as the histone code. So the deregulation of these epigenetic patterns can thus have severe effects on protein expression and alterations in epigenetic patterns are indeed observed in many cancers. Hence, it's one of the hallmarks. The figure here on the left shows a selection of known histone modifications in histone proteins H2A, H2B, H3, and H4. Neither the list of amino acid positions nor the possible modifications here is exhaustive. So only shown here are single methylations in yellow, acetylations in blue, ubiquitination in purple of several lysine and arginines, as well as in orange the phosphorylation of a few serines and tyrosines. Histone 3 alone has more than 30 known modification sites, most of which can have various modifications. So a complete figure would have would have not fitted in this slide here. So in the table here on the right, you can find a list of selected histone modifications and epigenetically relevant enzymes, and some products offered by antibodies-online against these antigens. So mono and trimethylated histone H3K4 are conducive to gene transcription. Trimethyl H3K4 is typically enriched at active promoters near transcription start site. Likewise, H3K9 acetyl is a mark for active promoters. Trimethylation of lysine 27 of histone 3 is considered a repressive mark, whereas trimethyl H3K27 is characteristic for polycomb silencing through the polycomb repressive complex. Acetylation of H3K27, on the other hand, is considered to be a transcriptional enhancer. H3K36 trimethyl then prevents aberrant initiation of transcription and is also implicated in DNA damage response and the recruitment of components of the DNA damage repair machinery. These histone modifications are carried out by histone deacetylases or HDACs and acetyltransferases or HETs, as well as histone demethylases and like, for example, KDMs and histone methyltransferases or HMTs. Antibodies-online offers too many products here for these enzymes and modifications to list them all here. So again, please get in touch if you need any products beyond what's noted down here. The RNA polymerase 2 at the bottom here, in the bottom row here, on the other hand, can only bind to open chromatin, and it's therefore frequently used to detect actively transcribed genes. So I hope to have provided a useful overview of the hallmarks of cancers, admittedly a bit fast, but yeah, there was a lot to put in here. But I still hope that it helped you to appreciate the significance of some of the essential proteins. To conclude, I would like to bridge to another related area of research, because a similar concept to the hallmarks of cancer was proposed in 2013 to summarize the processes that characterize aging. The first iteration of this list of age-related changes in physiology depicted includes the list of the represented list here, and you see, for example, that things like genomic instability, epigenetic alterations, and actually sound very similar to the hallmarks of cancer. So it becomes clear that there are common themes in both cancer and aging-related processes. So this is just an example, and yeah, I encourage you to, in general, be attentive to cross over between these two areas of research. Yeah, so this concludes the presentation part of the webinar, and so we can cover some questions that you might have right now.